Okay, all right, here we go. Good afternoon, everyone. So just a couple of things before we dive here into the lecture. So the first thing is to remind you that our first term quiz is coming up. So next Monday and Tuesday, the quiz will be available to you on Quarkus. So don't forget to log in at some point to complete that quiz. You have an hour to do it once you open it, and there are 10 multiple choice questions that you're answering. I'm going to put some channels onto the Discord server to facilitate any collaboration you want to do there. The only thing that we have to establish is that you can't actually post the question onto Discord. The reason being, obviously, if you wake up at midnight on Monday and post all the questions, then you're basically giving everybody 48 hours to sit around thinking about them, and you're kind of putting yourself then at a tremendous disadvantage. So you can certainly use the channel, use the Discord server to talk about the questions and talk about the ideas that you want to discuss related to it, but just don't post the actual question itself, okay? So don't forget about the quiz, that's number one. Once the quiz is done, so sometime Wednesday, I'll release the grades, and you can see how well you did. And then that kind of starts us into a 3-day period of potential rebuttals. So I'll post the instructions for how you rebuttal if that's something you want to do. But that'll then take place kind of the Wednesday, Thursday, up until the end of the day on Friday. And then lastly, with respect to short communications, so everybody should be in a group now. I put everybody in a group yesterday. So if you are newly into a group, meet your group mates at some point, reach out to them. Yeah, now is the time to kind of start thinking about aspects of physiology that interest you, research questions you might be interested in. Now's the time to start watching those module videos, figuring out what needs to be done, and starting to kind of chip away at this project, because it will take some time to get through it. There was a question over there. What was the question? Yeah. Yes. No. So the audio recordings that you see for some of the questions, those are just meant to be kind of helpful to you as you're kind of figuring out, like, how would I even approach this question in the least? So yeah, there won't be any audio recordings on the term quiz itself. They're just there to be a supplement to you while you're trying to learn how would I try to tackle these questions. Any other questions about things? Okay. I have a question, which is, I don't know, this morning I had to change. Did you like the yellow pointer better than the purple? Yeah? No? Like the purple better? Okay. All right, someone likes the yellow. Okay, well, every once in a while it's nice to change it up. And the program had updated and I had to re-upload things, so I just went to yellow. But you'll probably see some different colors maybe. Some like it, some don't. Okay, in any case, we're going to pick up where we left off on Tuesday. And we left off on Tuesday talking about, how can we create, antibodies for all these different potential antigens that we could be exposed to, that we can't possibly predict a priori what's coming at us. And so we want to have this broad coverage in terms of the kinds of antibodies we can make, the kinds of antigens we can recognize. But again, thinking about antibodies as being proteins, if we're trying to create, trillions of potential antibodies, how do we do that without having to have trillions of genes in the genome, which would, of course, be, unwieldy? So we talked about how the antibody genes are structured. So we have, just a handful of antibody genes, really, and based on their kind of segmented orientation, we're then able to create a very large multitude of antibodies from a relatively small number of antibody genes. And so just to kind of recap what we had discussed at the end, we had said that if you look at the structure of a typical antibody gene, it's divided up into many so-called V or variable segments, a certain number of D or diversity segments, a certain number of J or joining segments, and then you have the constant region exons, which of course are coding for the class of the antibody, IgA, IgD, IgG, etc. And so what we said is that when you're trying to build one of the light or one of the heavy chains that you're going to use to build your antibody, what you're going to do is you're going to pull just one segment from each of these three major groups. So grab one V segment, grab one D segment, grab one J segment, and put those together, and those will then constitute your variable region, right? And of course, thinking back to the structure, right, you've got two light chains and two heavy chains, so you're doing this, at least 4 times over to then have all the chains you need to build your antibody. And so because you can put these different V and D and J segments into so many different configurations, and then on top of that, because you could put a couple different, build your antibody from, this one light chain gene or this other light chain gene and this one heavy chain gene or this other heavy chain gene, by the time you put all these different segments together per heavy or light chain, and then by the time you put together your different heavy and light chains, you can actually create quite a multitude of antibodies starting from a very small number of antibody genes. And so this is probably a very liberal estimate, but some people have estimated that we can generate up to 50 trillion different antibodies from a relatively small number of antibody genes because of this segmental arrangement of the genes and how we are putting, again, just segments of the gene together in different combinations. We have, as you can see in the table, it illustrates there for us how many variable and diversity and joining segments we have for the various light and heavy chain antibody genes. So again, from a very small number of actual genes, we have a way to create a very large multitude of antibodies. Plus, I mentioned at the very end of the lecture that on top of this, if there is any mutation that occurs to the antibody genes throughout your life, this actually is a good thing, right? Because it now means you're going to create even more genetic diversity within your antibody genes, and this is going to give you the capacity to create new antibodies for new antigens. So yeah, mutation is actually kind of a welcome thing when we're thinking about antibody genes. We often think about mutations as a negative thing, and in many other genes in our body they are, but in the context of our need to just make as many different kinds of antibodies as we can to provide protection against all these potential antigens that could exist out there, mutation in the antibody gene is actually beneficial. Now, the next point to mention here, we didn't talk about this on Tuesday, is obviously if you're taking these antibody genes and you're building all these 50 trillion different antibodies in the way that we've talked, you can imagine that you might create antibodies that would actually bind to proteins that are actually in your body, right? What we call self-proteins, right? Proteins that are supposed to be in your body, functional proteins that we need to live and grow and survive and reproduce. And so the key here is that whole education process that we are going to put our immune cells through, right? Our B cells and our T cells, especially when we think about B cells and antibodies, we have to basically expose our developing B cells to all the different self-proteins in our body, and if any of them are making antibodies or B cell receptors that recognize our own self-proteins, then we need to kill them off, right, as a way to prevent the immune system from attacking our own healthy body proteins. And so that kind of comes into the next slide that I have here, kind of talking about how the immune system does this kind of education process initially. And so if we look at this figure here, we can start on the left-hand side with what they're calling a stem cell. They're not telling us whether this is the hematopoietic stem cell that gives rise to all blood cells, or whether, say, it's just the common lymphoid progenitor that gives rise to the B and T cells and natural killer cells, but it doesn't really matter which it is. The point is that that stem cell is going to give rise eventually to, in this case, a whole bunch of different B cells. You can see here they're showing us 8 different B cells that have come from that one stem cell precursor. And if you look at, in this case, the little B cell receptors that they're showing on the surface of all these B cells, and the antibody that those B cells would make would be identical to that B cell receptor, right? They need to be able to, whatever antigen your B cell receptors are recognizing, you're going to create antibodies that recognize the exact same antigen. We've got here these 8 different B cells, and each has put their antibody genes together in a slightly different way to create these different B cell receptors and therefore these different antibodies they could produce. And what you notice is that some of these cells have little X's next to them, and that's because during the education process, so while these B cells are hanging out in the bone marrow, we're going to expose them to all the various proteins in our body, and it was number 3, 5, and 8 that actually reacted, right, showed a reaction to our own self-proteins. And so the X here represents that those B cells then were killed, right? And so this is referred to as clonal inactivation. We are inactivating those B cells that have shown a propensity to react against a self-antigen, right? A protein that is supposed to be in our body that we do not want to be making antibodies against and trying to eliminate. So when I was talking earlier about the education process that these cells go through, this is what it entails, right? It's about taking B cells and T cells as they are maturing, exposing them to all the various proteins in our body, and then any that show a reaction to those self-proteins are ideally going to be destroyed. If they're not destroyed, then that could lead to the eventual development of some kind of autoimmune disorder. So what you're left with at the end of that education process, in this case, is the 5 B cells that did not respond to a self-antigen, and here they describe this as our repertoire. So this is now in this case, this very simplified case, these are the 5 B cells that are going out into our body. They're going to make their way into our lymph nodes, for example, and they're going to be waiting there, I guess in the hopes, maybe, that they will be exposed to the antigen that they recognize, and they'll have a chance then to actually respond. Yeah, the ones that have been X'd out here and did not make it into the repertoire were the ones that have been destroyed because they reacted to some protein that's naturally found within the body. Yeah, okay. Okay, so then we have our repertoire, and what they're showing us next here in the figure is that this little red antigen here, which nicely fits into this B cell receptor on number 2, we've become infected by it, right? So it's maybe an antigen from a pathogen, and so now this number 2 B cell has been exposed to the antigen that it recognizes, and so that's going to be leading eventually to its activation. And we start seeing that one of the things that happens as these B cells activate is they begin to undergo cell division. And so now we get what's called clonal expansion, where this one particular B cell, number 2, that has now been exposed to the antigen that it recognizes, it's expanding in terms of its numbers. It's undergoing mitosis to create more copies of itself, and that, of course, is going to give us a greater capacity to actually produce the antibodies that would bind to and help us to get rid of this particular antigen and its associated pathogen. So that's the expansion process. And then you start to see that little X's pop up next to some of these newly created B cells, and it says that they are dying. This death is by apoptosis. So it's a programmed cell death. It's an intentional cell death, right? And so again, as it comments here, and this has become one of our themes in this lecture, is we don't want to just expand out our B cell population, and keep it big forever and ever, because then we risk the over-activation of the immune system. We run the risk that we are creating so many antibodies for this antigen that we found that maybe that antibody will bind to something that's kind of similar, but is maybe one of our own proteins, and suddenly again our body is being damaged. So these B cells will eventually start to undergo apoptosis, right? They will start to die off, and the antibodies they produced will continue on for a while. But they also notice here that there are some of these cells that do persist. And they are essentially going to become our memory B cells. And what they're trying to illustrate here is that where we started before the antigen was here, we started with one B cell that recognized that antigen. At the end of this process, we have 3 times as many of these B cells that recognize that same antigen. And so consequently, when we are exposed to the same antigen again in the future, we're going to have presumably 3 times the capacity to respond to it, both in terms of speed and in terms of the number of antibodies we can produce. And hopefully that's going to mean that we can eradicate this pathogen much more quickly, the subsequent exposure, and then we're less likely to actually experience any kind of symptoms from that pathogen infection. And so one of the things, again, I want to drive home here, because I mentioned it earlier in the course, but it's a good time to remind us about it, is that, you know, the legacy of the first infection here with this antigen is not only going to be that we might have these antibodies that have been produced that are going to persist in our body and help us to recognize and eradicate this pathogen going forward, but we're also going to have these memory B cells that are left behind too, right? That if they are exposed to this antigen again, they can reactivate and become antibody-producing cells as well. So this got highlighted a lot during the pandemic, during the COVID-19 pandemic, because there was such a focus on, you know, we've given you this vaccine, your body made antibodies to it, oh, 6 months later we can't see those antibodies anymore, we better booster shot you. People were forgetting that just because there aren't antibodies right now doesn't mean that the capacity for the body to produce those antibodies from the memory B cells that are left isn't still there. Right? So we have to remember there's 2 kind of components here. There's not only the antibodies that are produced to provide some lasting immunity, but also the memory B cells themselves, which can be reactivated to start producing antibodies again in a subsequent exposure to the pathogen. The last point to make at the very top of the screen there is that based on what we've just talked about, you wouldn't be surprised then that the relative abundance of the different kinds of B cells that each of us has in our bodies is going to depend then on the particular antigens we've been exposed to over the course of our life. And that's what we mean by the term adaptive when we're talking about the adaptive immune system, is that this component of our immune system is variable among individuals and reflects the history of each individual's exposure to pathogens and antigens. Right? So if we could look inside me, for example, and look at all of the different kinds of B cells I have, they're going to partly reflect the fact that, okay, I grew up in, you know, rural southern Ontario, right? And so the kinds of pathogens and antigens that I would be exposed to are very different than somebody who maybe grew up in, like, you know, urban India or something, right? So every one of us is going to have a different collection, a different relative abundance of these different B cells, right, based on the kinds of antigens and pathogens that we've been exposed to over our life, okay? Yeah? Mm-hmm. So you could. I mean, she's asking, you know, basically, like, where's the balance kind of here, I think, between, like, the antibodies and B cells. And I don't think I have an answer in terms of, like, is there an optimum here? I think the point is that we don't really want, I mean, in general, I don't think we want to have too many of either, per se. If you have way too many antibodies being produced against an antigen and they're persisting, again, that just increases. I mean, all of the antibody-antigen interactions are just at fundamentally chemical interactions. So, you know, although we're trying to build antibodies that are very specific to a particular antigen, there could be cross-reactivity, right? Something that looks similar, we could bind to it and then react against it. So, you know, we don't want to have an abundance of either antibodies or memory B cells per se after the infection. We want to have a little more than we did before, right? The basic idea being, okay, before we got exposed to this antigen, we didn't have a clue it existed. Your body didn't even know, your immune system didn't know this antigen existed. Now it knows it exists because it's responded to it once. Let's just gradually boost the capacity up a little higher now for that antigen. So the next time we see it, good odds we will because it's out there somewhere in our environment, we're going to be a little bit better prepared to deal with it. But we're not trying to boost either of these components to such a high degree that there could be the chance of them responding to other kinds of more benign antigens, and then we might get, you know, autoimmunity or allergy or whatever. Does that make sense? Okay. Okay. Kind of dwelling a little bit more on this idea of memory, I want to show this slide here, which kind of illustrates for us kind of the first and the second response to the same antigen. And again, kind of focuses on these two antibodies that we talked about in class on Tuesday. So you can see here that when we have our first exposure to an antigen, we are eventually going to start seeing both IgM and IgG antibodies get produced, and perhaps IgAs and IgEs too. They're focusing on the two kind of blood-borne antibodies here in this figure. But the first thing you'll notice here in the primary response is that there's a bit of a lag here from when you are actually first exposed to the pathogen to when you start to see a response from your adaptive immune system in terms of the IgM or IgG production. And so that's typical of the adaptive immune system, that the first exposure is a slow response, okay? Keep in mind, of course, that during that whole time where there's a slow response from the adaptive immune system, the innate immune system is doing its thing, right? It's going to have detected this antigen. It's going to have launched, you know, all the different things that we've talked about, inflammation, fever, whatever. All those things are happening. There's just a bit of a delay before the adaptive immune system finally kicks on. You'll also notice here that the IgM antibodies are the first ones that are being produced, and the IgGs come later. The reason that IgMs come first, there's a couple of reasons. One, as we learned earlier, they are the default antibody, right? There's no specific cytokine signaling that is required for their generation, so they're the kind of default that get produced. They have, as we saw in the lecture yesterday or Tuesday, they're pentameric. So they have these like 5 different kind of, well, 10 really. Each one has 2, right? You get the Y shape to it. You have 10 different binding sites for antigens to bind onto. So in the very initial stages of an infection, having these kind of pentameric IgMs that can just, you know, bind up tons of antigens is a good thing. And it also mentions here that they have a relatively low affinity, which means they're not necessarily too specific to the antigen that they've been produced against. And that means that there's also a chance they're going to bind up to things that are just kind of similar to that antigen. And again, that's not a bad thing in the early stages of an infection, right? I mean, again, I'm trying to anthropomorphize things here a little bit, but you know, if you were told that, you know, this specific person here is a threat to us, right? Well, again, human nature would say, okay, well, let's just kind of look for anybody that looks even remotely like her, and let's just get rid of them all, right? I mean, I'm not advocating for cultural genocide in any way here. I'm just saying, like, you know, that's kind of the way we often do things when you think about wars. It's the way the immune system works. It basically says, she's my target, but anything similar in the early stages, let's just get rid of it. So that low affinity, that low specificity is kind of meant to be an early mechanism to help us get rid of anything that's kind of possibly a threat, okay? We also, and I added this point because, I mean, I guess over the years it sort of disappeared from the slides, but the IgMs are also way better at activating the complement system. So that classical pathway where the C1q has to find an antibody-antigen complex, the C1q responds way better to IgMs. And so having them produce first is going to help you to kind of get that complement pathway kicked on too. And then lastly, it mentions here how they're too big to enter tissues. They're pentameric, they're giant, they can't really easily get out of the blood vessels and get into our tissues. And so they're kind of restricted in terms of just being stuck in the blood vessels and really kind of looking for pathogens there. But then eventually the IgGs start to come in. Right, eventually we switch over once the helper T cells start signaling that we should switch over to IgGs. And so we eventually get the IgGs being produced and they kind of take over. And again, they are much more specific. They have a much higher affinity for the antigen. They're much more specific. So now we're narrowing down and saying, okay, let's just not attack everything that looks like it could be suspicious. Let's narrow down on the very specific identified threat. Okay, so we have this transition of our immune system from let's just try to get rid of anything that looks suspicious to let's focus on the particular identified target here. And then also, because the IgGs are monomeric and way smaller, they can actually get out of the blood vessels and get into the tissues. And now we start providing coverage to the tissues in terms of helping them to eradicate the pathogens they find. So that's what you see the first time, right? A rise and fall of IgM, followed by a rise and fall of IgG. The second response to that same antigen looks a bit different. I mean, the IgM response is pretty much the same, although it's now occurring much faster, right? You've lost that delay. So this is one of the aspects of the adaptive immune system, is that subsequent exposure gives you a faster response. And although the IgM response is pretty much the same, note how the IgG response is very much enhanced. It's a much larger number of antibodies that we're producing. They will persist for a much longer period of time. So again, this is part of this memory aspect of the adaptive immune system, that we are responding faster and to a larger degree to this specific threat, this specific antigen during the subsequent exposure. Okay. Okay, next I want to talk a little bit about, how exactly do antibodies protect us, right? I think sometimes we are very much aware that antibodies do protect us, and we talk a lot about antibodies, but what are the specific mechanisms? Like, how exactly do they work? And so the figure here tells us that there are basically 5 mechanisms, 5 different actions that antibodies do to protect us from the pathogens and antigens that they are being produced against. So I'm just going to go around the horn here. The first one is this agglutination mechanism. So again, what we have to appreciate is every one of these antibodies has that Y shape to it, has these 2 variable regions on it, right, that can both bind to the antigen or the pathogen that these antibodies recognize. And so what they're trying to show us here with this agglutination mechanism is how a single antibody can bind up 2 pathogens, 2 antigens, and kind of cluster them together. Or they're also showing us how here we have 2 antibodies kind of working in tandem to cluster 3 separate proteins, 3 separate pathogens rather, into a single cluster. So the idea of agglutination is that instead of having a whole bunch of individual pathogens and antigens out there, the antibodies help to cluster them together into these larger but fewer number of units. And as it mentions here, this is advantageous because if the phagocytes are going to come along here and eventually eat these pathogens up, they now only have to worry about like these 2 entities to eat as opposed to the 5 original entities that were there, right? So it's like you're trying to kind of cluster your meal into these like, bigger, fewer but bigger bites kind of a thing, right? It's like if you imagine that you had to eat your rice individually, 1 grain at a time, right? That's a real slow process of eating rice. But if you have the spoon that kind of clumps the rice together, or a sticky rice or something that clumps it together, now I can have many more rice per bite I take. I'm going to be able to eat that rice a lot faster, right? And so if we assume the goal is to eat rice as fast as possible for whatever reason, clumping the rice together into fewer bites but larger numbers of rice consumed per bite is going to be faster. Same thing here. If we can clump our pathogens, our antigens, into a fewer number of units that the phagocytes have to eat, they're going to be able to eat them up and eliminate them at a faster rate. So that's the agglutination, that antibodies will cluster pathogens together to help phagocytes have bigger but fewer things to eat. Second here, and we've talked about this quite a bit, is this opsonization idea. So again, that word opsonization, opsonin, I think again it comes from Greek, I believe. It means to make tastier. So we're talking about how antibodies will make a pathogen look more appetizing, essentially, to a phagocyte. And so the way this works, and we talked about this a little bit before when we were talking about complement proteins. We said, hey, that the C3b, that complement protein that coats a lot of pathogens, phagocytes have a CR1 receptor on their surface that binds to that C3b. So we coat the pathogen in C3b, and then that makes it more likely to bind to that CR1 receptor on the phagocyte surface, basically helping to bring the pathogen to the phagocyte to be eaten. It's the same thing here with antibodies, because on the surface of phagocytes, they have what's called the Fc receptor. So this binds to the constant region of antibodies. So you coat your pathogen in antibodies, and then the antibodies bind to the Fc receptors on the surface of phagocytes, and again, that makes it easier for the phagocytes to interact with these things they need to eat and get rid of. So this is very similar to that CR1 mechanism we talked about before. The third mechanism here is neutralization. So many pathogens will use the antigens on their surface to gain access to our cells. Right? You think about COVID-19, there was a lot of talk about the ACE2 receptor, right? There's a protein on the surface of the COVID virus that binds to the ACE2 receptor, and that triggers it to be internalized into the cell, and then it infects. Well, if you can use an antibody to bind to the surface of that receptor, or sorry, bind to the surface of that antigen on the surface of the COVID virus, so that now that protein can't actually interact with that ACE2 receptor, then you've basically kept the virus out of the cell. And obviously in the case of a virus especially, if viruses can't get into the cell, they can't replicate, they can't do anything, they're not going to cause infection. Same thing with bacteria, same thing even as I mentioned here with things like bacterial toxins. You know, a lot of bacteria make us sick because of the toxins that they secrete into our body. You know, and these toxins exert effects on our body in some way. If the antibodies can bind up to those toxins and basically block them from binding onto things in our cell or on our cell surface or wherever, then ultimately those toxins can't exert their toxic effects. So that's what I mean by neutralization. It's basically using the antibodies to coat and cover up the pathogens, the toxins they produce, to prevent those things from being able to use their antigens to interact with our cellular components. Number 4 down here in the bottom right corner, it talks about antibody-dependent cell-mediated cytotoxicity, which is a kind of a mouthful, but what they essentially mean here is that antibodies also bind onto natural killer cells too. Because natural killer cells, which we've talked about before, they also have these Fc receptors on their surface. And so when you have an antigen or a pathogen coated in antibodies, not only does it make it easier for them to bind to the phagocytes and be eaten, but it also attracts them to the natural killer cells. And then the natural killer cells can inspect them and go, oh shit, I don't see an MHC class 1 protein here perhaps, I'm going to kill this thing, and it's gone. So you're basically enhancing the natural killer cell's capacity to do this immunosurveillance that it does. And then lastly at the top here is the activation of the complement system, which we've already talked about, so I don't want to rehash too much here. But we know that the end result of that process is the formation of that membrane attack complex that gets embedded into the cell membrane of the pathogen, and then essentially you poke holes in the pathogen membrane and it basically dies as a result. Well, what they're also showing us here is that as those pathogen cells are lysed open by this complement system, of course they're going to start leaking out all of their cell components. They're going to leak out any pathogens that they had inside of them, or sorry, they're going to, not inside of them because you're hitting the pathogen, you're going to leak out all these pathogenic proteins. You're busting up these pathogens, you're going to be releasing all these pathogenic proteins and all the stuff that was inside the pathogen into the body. That's where the inflammation response comes in here, right? That as all these pathogenic proteins are released from the pathogens as they die, that's going to actually trigger an inflammation response. It's going to trigger resident macrophages to detect these things. It's going to trigger the recruitment of neutrophils and monocytes to the site to help eat up and eliminate all those pathogen-derived proteins that were suddenly leaking out as you kill the pathogen. So yeah, those are your antibody mechanisms of action. So antibodies are doing a lot of stuff as part of their mechanism to again, protect us from the antigens and pathogens that they recognize. So we're going to move away from the B-cell immunity now and shift over to T-cell immunity. And so I'm going to talk first about cytotoxic T cells. These are also known, as you can see there, as CD8 plus cells. And that's because, as they illustrate here, there is a CD8 receptor that is on the surface of these cells. So let's start there. What does that CD8 receptor do? Its job, as again they're trying to illustrate here, is to basically interact with the class 1 MHC protein that is on the surface of our body cells. So again, we've learned about the MHC proteins. There's the class 1. These are the proteins that are expressed on all the cells of our body, with the exception of the antigen-presenting cells, because the antigen-presenting cells use an MHC class 2 instead. So CD8, its job is to look at this cell that we're bound to right now and basically assess, is there a class 1 MHC on the surface of this cell, or is there a class 2 MHC on the surface of this cell? Because T cells want to kill only cells with class 1 MHC. They don't want to kill cells that have class 2 MHC because those are antigen-presenting cells. Those are B cells. Those are cells that are trying to alert the immune system to, here's the invader I found. So you don't want to kill off the cells that are presenting the antigens to try and activate the immune system. You just want to kill cells that are infected by the antigen. So the CD8 is going to identify, is this cell showing me a class 1 MHC? If it is, then we could go ahead and kill it if it's infected. If what it's showing me is a class 2 MHC, we're not going to kill this thing. This is an antigen-presenting cell. We're not going to kill it. Otherwise, that's actually going to be a bad response for our immune system. So that's the primary job of the CD8 receptor. It's that MHC class distinction. So if CD8 says, yes, this is an MHC class 1, then we turn our attention to the T cell receptor, and it's going to actually inspect the antigen that's being presented on that MHC class 1. And if the T cell receptor says, hey, I recognize this antigen, right? This shouldn't be in the body. And if the CD8 has said, yep, and this is an MHC class 1, so this is a killable cell for us, if those two things both pass the inspection here, then we're going to kill this cell, because it means that it's an infected cell. It's not an antigen-presenting cell. It's a cell that's infected. We want to kill it. It's a cancerous cell. We want to kill it. And so once you have those two checks, CD8 check, TCR check, then the cell will release these two proteins. One is perforin, and then the granzymes. Releasing the perforin, what it does, it's the blue protein here, is perforin actually embeds itself into the cell membrane along with other perforins to create these pores in the cell membrane, to create holes in the cell membrane. Now we've said that that, in part, would kill a cell because you've now disrupted the structure of its plasma membrane. So that's part of what it's trying to accomplish, but then also there are these granzymes that are released. These are proteases. And they're now able to enter the cell we're killing here through these pores that we've generated through the perforin. And so the granzymes go into the cell, and they start chewing up all the proteins that are in that cell, and that, of course, will definitely kill the cell. Right? Proteins do all the cell stuff. So if you chew up all the proteins in a cell, it's going to die. So you had this CD8 and TCR inspection, check, check, release the perforins to create these pores, release the granzymes to enter through the pores, chew up all the proteins in this cell, kill the cell off. Okay, so now you see our CD8 cell is released. It's going to go on and inspect another cell. It's left this target cell just to die on the street corner, right? But that's fine. It was an infected cell. We wanted to kill it anyways. Again, just as we were talking about with antibodies a second ago, you might be thinking, well, I mean, there's going to be the release now of whatever was infecting this cell. As the cell dies, all of its contents are released. So just like we were seeing there a moment ago with the lysis that's caused by the complement system, basically we're now relying on inflammation, the inflammation response, to come in here and say, okay, I see all these pathogens that have been released from this dying cell. I've detected them. I can eat them. I can recruit in some other neutrophils and things. The inflammation response basically is going to take over at this point and help to clean up the debris left behind here by the killer cell, right? So it's kind of like, you know, an assassin coming in, pew, pew, and then there's a dead body, and then somebody else comes in and is like, okay, well, I got to clean up this mess because we can't just leave dead bodies lying around. spewing out infection or something, right? So it's kind of a team effort of sorts. Now, one of the questions that I think is worth thinking a little bit about here is, if you're a killer T cell, a CD8 plus cell, and you're releasing the perforins and granzymes to try and kill the target cell here that's infected, how do you not die in the process, right? You're releasing these proteins into your vicinity. Why doesn't perforin come into your membrane and make pores in it? And then the granzymes come in and chew you apart, and now, you're dead. Well, we're going to talk a little bit about that. There's a kind of a clever mechanism at play here to stop that from happening. So before I show you the data here that illustrates that clever mechanism, we have to just look at this y-axis here for a second. So the y-axis says that they are measuring chromium-51 release. So why? What is this all about? So this is a very common method used when you're trying to figure out the ability of something to kill cells. So what you do is you take whatever target cells you're interested in studying, right? Whatever target cells you want to see how well they can be killed by something, and you soak those cells in a solution of chromium-51. This is a radioactive version of chromium. So you soak the cells in chromium-51, and the chromium-51 is able to enter into the cells. As it comes into the cells, it actually gets reduced, right? So it goes from being chromium-4 to chromium-3, and then in that chromium-3 form, it actually can't get back out across the cell membrane. It becomes cell membrane impermeable. So you're soaking the cell initially in this chromium-51, chromium-4 version, right? This oxidized version, shall we say, of chromium-51. It's cell membrane permeable. It can come in and build up within the cell. Then as it's in the cell, it gets reduced into this membrane impermeable form, and now it's trapped. So you end up with these target cells you've got all filled with this radioactive chromium that's trapped inside and can't get out because it's membrane impermeable. And then you basically mix it together with whatever thing you want to see if it can kill it. So maybe you mix it with a natural killer cell, or you mix it with a cytotoxic T cell, or whatever. And then, if that thing can kill the cell, well, then that will let the radioactive chromium leak out of the cells and into the surrounding fluid. And then all that you have to do is you have to take that whole mess there, centrifuge it down, right, so that you just end up with this supernatant fluid here, and then you measure, well, how much chromium-51 is in there. Right? It says you're going to use this liquid scintillation counter. It's basically a gamma ray counter to measure the radioactivity of that chromium-51. And so the more cells that have died, the more T cells, or the more target cells that had this chromium that died as a result of exposing them to whatever killing mechanism you were looking at, you're going to be able to say, well, the more radioactive chromium that leaked out of these cells and into my supernatant here, the more death of those cells that occurred. Okay, so when they talk here about measuring the chromium-51 release, they're basically measuring how much cell death occurred, right, to these cells. Now there are 4 cell types that they examined here in this study. The first 2, it says, these were cultured tumor cells. So they were cancer cells, right? You'd want to know that, your T cells could kill off cancer cells. And then the other 2 here are the natural killer cells and the CTLs, the cytotoxic T cells. Now, I'll just mention here, because I don't think we talked about it yet formally, remember how natural killer cells, they are derived from the same precursor as the cytotoxic killer cells, right? That the natural killer cells and the T and B lymphocytes all come from that common lymphoid progenitor. So natural killer cells, when we talked about them killing the cells that don't have MHC class I, they kill by the same mechanism as these cytotoxic T cells. It's this perforin granzyme-based mechanism. So that's why they're kind of examining both of these types of cells here in this study. So we've got our 4 different cell types here, our tumor cells, and then our immune cells, our cytotoxic T cells, and our natural killer cells. And what they're doing here in this study is they're exposing these cells to different concentrations of perforin. And then they're assessing how well is that perforin able to basically permeabilize the cell membrane with pores to allow that radioactive chromium-51 to leak out, right? How effectively can perforin damage these cells? And you will notice here that the two tumor cell types, right, they start beginning to die at fairly low perforin concentrations, but notice that it takes way higher perforin concentrations to kill off the cytotoxic T cells or the natural killer cells. And I'll just highlight here, this is a log scale, right? So every jump here we're talking is like 1,000-fold change. So we're talking about substantially more perforin resistance in our natural killer cells and in our cytotoxic T cells relative to the cancer cells that they're trying to kill off. So this is how these cells try to prevent themselves from dying. This is how the cytotoxic T cells and natural killer cells resist their own death mechanisms, is that they have become perforin resistant, right? They can't have holes poked in them by the protein they're releasing. But that, of course, then just begs more questions, which is, well, how do they stop that perforin from getting in there to permeabilize them and kill them? And it turns out it has everything to do with their unique phospholipid composition of their cell membrane. This beautiful image here, so these are, they might be freeze fractures or they're scanning electron micrographs, I'm not 100% sure, but you can see here that they're looking at perforin and they're looking at pure membranes made of just this one particular phospholipid called phosphatidylcholine, or PC. And then in the bottom, they're looking at perforin embedded in a pure membrane of phosphatidylserine, or PS. And what's very abundantly clear from these images is that when you have the membrane with lots of this PC, this phosphatidylcholine, the perforin proteins are able to organize themselves into those beautiful pores that are going to let stuff, let granzymes get in to kill the cell. But when you have a lot of phosphatidylserine in your plasma membrane instead, the perforins can't organize themselves into pores. And so what happens is that at the moment that these natural killer cells and cytotoxic T cells are releasing perforin because they've detected that they've got a target cell they need to kill, what these natural killer and cytotoxic T cells do is they very quickly change their membrane composition to add a whole bunch of phosphatidylserines to their membrane to prevent the perforins from being able to organize themselves into pores that would then permeabilize the cell membrane and allow granzyme to come in. But this is a tricky balance too, because when you have a lot of phosphatidylserine in your plasma membrane, that also is actually a signal to the immune system that this cell is fucked up, we better kill it. So they have to do this kind of real quickly, like change the membrane, release the perforins, protect themselves for a bit, let the cell die, but then switch back to a more PC-rich membrane so they don't attract attention to themselves because of that abundance of phosphatidylserine. So again, as with everything in the immune system, it's this kind of delicate balance that's at play here. Okay, we will stop there and we'll pick up tomorrow. I'm hoping we'll finish immunity tomorrow. That would be the goal, so that we can start fresh on Tuesday with something different.